morning, one and all. Science Academies virtual lecture workshop on recent advances in microbiology and biotechnology, day 2. With getting the blessings of our Lord Almighty, let us pray for the successful completion of the day 2 webinar.
I
request the fellow participants to mute yourself. Shyam, JP sir.
I muted, sir.
Today, one of our eminent personality with us, Dr. T.K. Adya, who is the professor of the Kalinga University, Bhubaneswar, Odisha. I feel wholeheartedly thank you for his availability with us for enlightening our students. We would like this day 2 webinar with the moderator of our PG Association President and the Assistant Professor of Department of Biochemistry, Dr. G. Shivayalango. Before going to that, I'd like to thank our management and for the principal for permitted us to conduct this webinar in the centralized way for the benefit of our students. As our students are assembled in the Karina Hall, this much of numbers are assembled here to listen the expertise speech from Dr. T.K. Adya sir on his presentation. Now I would like to formally start this session with the request to Dr. G. Shivayalango to start this webinar with the introduction to chief guest. Dr. Shivayalango, please proceed.
Thank you, sir. Yeah, very good morning and warm greetings to technical session 5 of the workshop. With great honor and due respect, I feel privileged to introduce the resource person of this session, Professor Tapan Kumar Adya. Position, Professor, School of Biotechnology, Kalinga Institute of Industrial Technology, Bhubaneswar, Odisha. Sir started his career in 1973 as lecturer in a postgraduate government college. Later, he joined Central Rice Research Institute, Cuttack, as junior scientist in 1977 and rose through the ranks to become the director of the institute in 2008. After completion of his term, he joined the Kalinga Institute of Industrial Technology, deemed university, as professor in the School of Biotechnology in 2012. He is also worked as co-director science of the UKRI GCRF SANH project since 2019. He acted as consultant to several national and also international research programs. Using tropical paddy as a model ecosystem, the entire focus of research by SAR is an sustainable management of resource-poor tropical soils to maintain high productivity with minimum environmental impact. He has contributed to India's first NATCOM on GHG and was invited in the elite panel of reviewers of the IPCC 2012 database and methodologies 2013 on GHGs by WMO UNEP. He has contributed to integrated global framework to assess the current capability and future vision on the impact of land use and management change on soil carbon and prepared a UNFCCC document on standardized baselines for rice cultivation CAM methodology involving Annex 3 countries of Africa. His latest assignment as Director, South Asian Nitrogen Center, New Delhi, involves coordination of research work and policy implementation on reactive N in different sectors of economy of South Asian countries. He was worked on health and environmental implications of low carbon climate change resilient diets in India 2016 to 17, funded by Wellcome Trust UK as co-investigator. He is also coordinating a GEF UNEP funded project on integrated nitrogen management system INMS for its South Asia operations. Professor Adia has published more than 180 peer-reviewed journal papers with a total citation of more than 8,200 with an H index of 47 and I-10 index of 118. He supervised 18 masters and 15 PhD students. Professor Adia is the editorial board member of Journal of Indian Society of Social Science, Biology and Fertility of Soils, and editor for Indian Journal of Microbiology, Agricultural Research Journal, Proceedings of National Academy of Sciences, Biological Sciences, Frontier Journals, Microbiology, Health Sciences, Ecology and Environmental Sciences, Sustainable Agriculture. Associate Editor in Field Crops Research Elsevier, and editor in special theme issue of the Philosophical Transactions of Royal Society B. And awards: Samanda Chandrasekhar Award for the year of 2008 by Odisha Legium Academy under the agencies of Department of Science and Technology, Government of Odisha. Professor G. Rangaswamy Memorial Award in Agricultural Microbiology for the year 2009 by the Association of Microbiologists of India, New Delhi. Next, Dr. N.S. Randaswa Commemorative Award for Excellence in Research in Natural Resource Management for the year of 2010 by the National Academy of Agricultural Sciences, New Delhi. Next award, first recipient of ICR Norm Burlock Award, the highest award in the agricultural in the country, for providing a breakthrough for agriculture through a new insight that has created high potential value for future, for the year 2010 by Indian Council of Agricultural Research, New Delhi. Next award, Professor Krishna Sahi Bilfami Memorial Medal for the Venium 2011 to 13 for recognition of outstanding contribution in the field of agricultural sciences by the Indian National Science Academy, New Delhi. Thank you. Now hand over the session to Professor Adia. Participants are requested to type the questions in the chat box. Once again, thank you all.
Thank you, Dr. Sivalinga, for the nice introduction. I started working quite early, so maybe. This today's lecture I have picked up in two parts related to microbiology. First one is soil micro. In the afternoon session, I'll be speaking about plant microbiome. So soil microbiome, I mostly restrict myself into the biodiversity aspect. So I have given the title of soil microbiome, that is biodiversity under our feet. Now, it's not moving. Yeah. Now, soils, of course, from our childhood we know, and the young friends sitting in the audience and others, they must be, their memory is much fresh than ours. They must remember how the parents used to scold when they used to play with soil. So also had been in our case too. Because general concept is that soil is dirty and means all sorts of problem and other things. That's of course is a sweet memory. But coming back to science, I think soils are called the skin of the earth. They are complex mixtures of minerals, water, air, organic matter, and countless organisms. Organisms, I say here, that are the decaying remains of once living things. That means dead and decaying, that is what is called detritus, dead and decaying biomass. All these things constitute what is known as the soil. You can see in the picture, nice dark complexioned mass on the hand, on the palm. And this actually is called the skin of the earth, as I told earlier, and is the direct support for plant life and is vital to life on earth, because the soils follow, they support all the plants. Now, let us first see about the soil functions. European Commission in 2006, they have defined 7 soil functions. Like one is that I have listed them: biomass production, including agriculture and forestry. So whatever agriculture we grow and the forests they grow, they are all, this result into the biomass, plant biomass, and animal biomass also to certain extent. Especially this production of biomass is supported by the soil, then storing, filtering and transforming nutrients, substances and water. Water, it falls on the surface, then passes through the soil and then gets accumulated in the groundwater reserve or sometimes on the surface reserve. And the nutrients also are transformed in the soil, and these are done by the soil. Then the biodiversity pool, such as the habitat, species and genes. Genes, because sometimes several DNA fragments are available in the soil carrying some important genes, which I will focus subsequently. Physical and cultural environment for humans and human activities. Source of raw materials, acting as carbon pool, because they store the carbon and also release under adverse conditions, under certain conditions, they release carbon also. And this actually, so they act as the carbon pool. And then archive of geological and archaeological heritage. Obviously, all this archaeological heritage and the geological heritage that we find, this is all on the soil. So these are the 7 soil functions which has been listed. Now, soils does not mean that only dirt and clay and all sorts of things. They are also alive. You can see here picture of several organisms which means that they are present in the soil. You can see nice beetles and the nematodes, all those things are there. Now, this is a poster by the FAO, which has, I borrowed it from the FAO website. The soil biodiversity, the hidden world, and it is the beneath our feet. It's a separate world. In fact, you can see here on the right half of the panel that the plant is growing and there are animals, big animals, and the fungi, and this root plant sends the roots, and in the root, through the root, some compounds are released, organic compounds, and also they support the different organisms. Now there are different layers. There is a base rock, then there is the rock and soil mixture, and up you come to the soil where all these biological life persists. Now there are different soil communities present in the soil. I think here it is not very clear, so I'll go to the next slide where it is more clear. Now the soil biodiversity, that is biological organism, diverse biological organisms present in the soil. We try to group them in depending upon their functions. First, we call a group as the chemical engineers. They are no way less important than the chemical engineers we see in the factories, putting their hat and then moving in the factory, looking after the different chemical fermenters and other type of things. Chemical engineers, These chemical engineers, however, are the bacteria, fungi, microalgae, etc., etc. What they do? Decomposition of the organic matter and the availability of plant-friendly nutrients such as nitrogen and phosphorus. So they degrade the organic matter and take out the nutrients from that, supply it to the other organisms, including the plants, and the availability of plant-friendly nutrients, specifically the different mineral nutrients, etc., like nitrogen and phosphorus, they are supplied by this group of organisms. So that's why they are called as the chemical engineer. Now the micro predators, they are the predators. Just a minute. Yeah, they are the predators like nematodes and protozoa. You can see here protozoa and the nematodes. Different nematodes are here. They do different job, like they feed on the microorganisms. This protozoa here, they can eat up several bacteria. You can see here one organism there, the bacteria are inside the thing, and the nematodes also. The nematodes also feed on the fungi and fungal spores and the microorganisms. They are the micropredators. Litter transformers, these predators are, they are biggers, but what they do is that they transform the litters, like they are different bigger ones like Acaridae, Onicidae, then Columbolus, and the Myriapodas. You can see here nicely, Myriapodas, and the different insect and insect larvae. They are spiders, etc., etc. They are the litter transformers. Now come the ecosystem engineers. So the earlier one, both the litter transformers and the microorganisms, predators, and litter transformers, they're all the chemical engineers, because they transform the organic matter, they degrade the organic matter and then make them transform. And also some of them are have specialization in nutrient transformation. Now come the ecosystem engineers. Like engineers build the different structures, the ecosystem engineers like ants, termites, and earthworms have the possibility of to structure physically their environment by producing biogenic structures. You might have seen, some of you, termite mounds, nice big mounds the termites make, or the earthworm casings or castings, it is also called, in the soil, which actually are they result into the different structural changes into the soil. Earthworms, for example, they move through the soil, resulting into the aeration and thereby making the soil more healthy. So it is a common saying, the farmers will say, they will scoop up a handful of soil and then find that if the earthworm is moving in that, they say that the soil is very good. Because the earthworms, they move through the soil and they have got the burrowing habit, and then passes through the soil, make the tunnels through which the oxygen can move and allow the other organisms to thrive. Now, if we see it, this is small, I mean, not a very small one, but a big table where it shows that the overview of average local species richness and approximate maximum abundance of the different groups of organisms, key, soil, microbial, and invertebrate taxa. Now you can see here prokaryotes. Prokaryotes, the local species richness is around 100 to 9,000 per cubic centimeter. This way the different groups are there, the fungi, the other group of organisms, and nematodes, etc., etc. You can see the known species and the estimated richness, these last two columns. First one is the distribution. That means how much are present in per unit area, like prokaryotes, around 100 to 9,000 per cubic centimeter. But the known species is around 4,500. In case of fungi, around 100,000. And the estimated richness, you see, bacteria, prokaryotes, they can be up to 10 to the power 9, depending upon the soil properties. And in case of fungi, it can be 5,000, and then so also, where all the other group of organisms, a tentative estimate has been made that there is a large number of organisms, locals, they are present in the soil, and they lead to the global species richness. Now here it's a simplified soil food web, where you can see that how these organisms, they or the microbes, they interact in some of the key group of the soil biota, for they interact towards a simple soil food web. At one end, we have got the plant, the nice green plant with the roots going into the soil. And these roots, they release the root exudate, but in the root region, you have got the plant parasitic nematodes. Associated with the roots are the mycorrhiza. And these mycorrhiza being fungi, there is the fungivorous mites also will be available, who will eat up some of them. Otherwise, also there are fungi, and these also follow this thing. And the litter transformer, I was telling that different beetle and the necarids and the earthworms, etc. They work on the leaf litters, and then they will break down, and some of them will die and then result into the detritus, decaying biomass, which will help the bacteria to grow. Now there are also bacteriophagous nematodes and protists who will eat up some of these bacteria and result into their growth. And the other side, there is the predatory mites and the predatory nematodes. They also all grow. So these actually, they derive the nutrition by the mites. Predatory mites will eat on the other columbolas and all those things, and then they will grow. So basically, it's a simplified. Although it is not this simple. Sometimes there are several steps in between. But the soil food web, they will show different groups of soil biota. Now, the microorganisms inhabiting soil perform various functions. Several functions they perform and entail ecological interactions that influence the soil functions from micro scale and end up to the landscape scale. That means from a handful of soil to a large area of the ecosystem and landscape scale. Now, this microbial diversity and the associated functional traits, they are highly dynamic. I mean, it's not strict or restrict to certain limit. They are highly dynamic, both temporal and spatial. Temporal means in relation to time and in relation to space, either responding to natural factors or to human-induced perturbation. Human-induced perturbation, I think next slide, maybe we may be showing them. And then this shows that the soil, but the thing is that in spite of this much huge diversity, a comprehensive assessment of the soil microbiome is still one of the biggest challenges in life science due to its high complexity. You can't simply take it for granted that this organism is there, that organism is there, but the interaction and the association amongst them is the most interesting part of the study. Yeah. Now, this is some sort of a model which shows on the left hand side. Don't look at the blue portion. Now we are talking about the soil, that is the brown color one. Now these are the intervention by the anthropogenic perturbations like cultivation. You grow the crop, for that you disturb the soil, or sometimes we water the soil, flood it. For say growing paddy, you have to put water onto the soil. So all this anthropogenic perturbation, anthropogenic means man-made, and the natural is that normal this thing developments like overflowing the river, and then this Gomati river, and then all the water flows onto the side of the soil, and the soil gets flooded. That is natural. But when we allow water for growing the crop or the cultivation purposes, these are the anthropogenic perturbation. Now, this type of perturbation, they affect the several soil functions like ecosystem services, functioning of the soil, and the physico-chemical properties, they all intervene together to affect the soil biological properties. The functioning of the soil as a result of this intervention, the other ecosystem services, and they are all affect the biological properties, which are usually measured in terms of the microbial biomass, microbial activity, and microbial diversity. These are the three cornerstones of studying the biological property. This I will mention in the on the right hand block. So the rare taxa or species potentially play key roles in the rate they on the soil function. So the one thing you can see here, it's a block of it's a block, three-dimensional block is being seen, soil, but actually the soil represents the four-dimension things. Another one which is not shown in, cannot be shown in this picture, is the interaction between the different sectors or different segments, including the life forms and the non-life forms, to result into the overall functioning of the soil. Now, this, how do we study that this type of things? There are several studies, and this actually is the basic microbial ecology. There is also now molecular techniques are involved in this, and then so it is called the microbial molecular technology. But basically, when we were students like some of like you, then our time we used to just to be very happy to take a small sample of soil, prepare a suspension in water, put it into the culture medium, get some nice colored bacteria, nice forms of bacteria, and be very happy that these are the diversity. But even then, you could see on the plate different types of organisms used to grow. Now, the techniques, scientific techniques have increased, have been bettered to a great extent. And then these are the basic checklist. What we do is that you have to have a solid experimental design, if you are taking in the field testing, then number of replicates, then the real control. A control should always accompany your treatment. Like say you want to see the effect of flooding or the water addition, then you should also have another treatment where no water is added. So a typical control, a real control should be there. And then the observation from time points, you have to go on taking observation, then only you'll be able to know the differences. Then the sample storage, then extraction methods of the different extraction methods you follow. Now, among these things, we have got nowadays the technology has developed like anything, and then what we call as the omics techniques have come up. You are also acquainted with this definitely, like metagenomics, metatranscriptomics, and metaproteomics. And there is also another one is that bioinformatics, which integrate all these observations in statistical terms and then try to find out the interaction, overall interaction. Maybe to certain extent it helps to explain the fourth dimension of the soil. So these are the molecular approach dependency. Now I was mentioning there are different. One of the most stable thing is that stable isotope probing or SIP, which is currently the best techniques. The PLFA SIP or the protein SIP, DNA SIP, etc., etc. So these are the different techniques we use. But I tell you, no technology is perfect, and people are still developing the technologies because we don't have answer to all the questions that is raised while explaining the microbial diversity. Okay, now this is another approach where we are trying to show the composition, expression, and activity of the different microorganisms and the techniques involved with that. I specifically mentioned this portion because you people who are interested to take up research in future, they will be when you will be handling some of these techniques, it will probably ring in your mind that these were introduced to you in your course of study, about the different things, composition, and then we divide them into composition. How do we measure the composition? Composition we measure by either metagenomic studies or which are amplicon studies, take the amplicons and then clone them and then find out, or metagenomic shotgun method, whereby we take the DNA portion and then find out what are the base composition, and then try to assign them their genetics relationship, and then find out all the details. Now this there we now we have got the answerable. Once we do that, now let us see what are the questions that we are able to answer. The metagenomics, especially the amplicon sequencing and other things, will tell which microbes are there. What are the different microbes which are present in metagenomic shotgun method? Which microbes are there? Again, the same thing, but then what might they do? That is additional information we find. So you see that those days when you used to put the small sample of soil onto a petri dish containing nutrient medium and then isolate the bacteria and try to find out. Now afterwards we found that around 98% of the life forms are not possible to be analyzed by the plating. So different techniques have come, and these are some of those. And these techniques will give us metagenomics, where we try to find out the composition. They tell us which microbes are there and what might they do. But the challenges, there are several challenges, is that the correlation and abundance of the metabolomics observations is not done. Then draft metabolic reconstructions need curation, and then rare microbes need enrichment. Especially those which are which live on very low nutrient concentrations. They had to be grown separately. These are the different difficulties that are there. Now coming to the expression, okay, these are the microorganisms we found out, composition we have found out. Now expression usually we try to find out through the metatranscriptomics, whereby because the presence of a microorganism in an ecosystem or in the soil does not mean they are all active and they are doing all the functions, including fighting amongst themselves or pulling down others' leg. Not like that. Some of them may be sleeping, some of them may not be working at all. They feel lazy, they don't work. So these things can be found out only by studying the metatranscriptomics, whereby we analyze the transcriptome and then find out who are the active microorganisms, okay, which microbes are there, and then which microbes are doing what, whether it's a nitrogen fixing bacteria. So NIF gene is activated. Transcriptomics, we find out that the activation of the NIF gene, and then we tell that yes, these are the group of organisms which are fixing nitrogen, right? Then the metaproteomics, it is another step. We go ahead. Whatever the DNA is replicated, transcriptomics, we come to know, all of them may not be finally working as the enzyme or the structural protein. So that is known by the metaproteomics, whereby we extract the protein and then assign their role by different techniques are available, by assign their role by either the structural form or the functional forms. Yes, this is a cellulose enzyme which is functional in this forest soil, because the forest detritus, plant detritus, they are falling onto the leaf, dead and decaying leaf are falling onto the soil. So these microorganisms, they produce the cellulose enzyme, and this cellulose we can identify through metaproteomics, and which are there and they are functional. Now, still we can go one step ahead, whereby we try to find out the metabolite they produce, that is the activity portion. So first one was the composition, then was the expression, and third is the activity. We know the metabolites which are being formed, and these metabolites can be found, estimated, and then told that yes, these are the group of organisms which produces certain metabolites, and because of that metabolites, either they stop the other microorganisms to grow, or they help the plant to protect themselves from the attack of different pathogenic microorganisms. So these are all the different group of study. Okay. Now this, as I said, that these methodologies are being regularly developed and none of them are fully competent. So there are several methodological errors introduced by the analysis of soil microbiomes using, I will specifically mention about DNA-based approaches. This is because soils are 4D objects, and then usually in all our studies we take consider the only 3-dimensional issues for the soil. Second thing is that DNA extraction is the most ignored source of error. We extract the DNA, fine, very good. On a nucleic acid based analysis, we use a primer and then try to find out which whether that DNA based sequence is available in the soil or not. But this then another thing is that the marker gene based approaches almost primary. Here the primer you are using, and here you are using the marker gene, and this actually is not foolproof, because there could be a group of bacteria who, as we are all talking about, say for example, nitrogen fixation, NIF gene. There could be another group of organism which can also fix nitrogen, but by a modified genomic approach, modified gene function, we may not be knowing. So if we go on the studying only using the NIF gene primer, then we will be missing those. So those case of rare biosphere organisms we cannot study. So you have to go on approaching the problem by several ways. Not only that, okay, you have extracted the DNA with the nucleic acid based analysis or the marker genes you are trying. So that can not give us 100% knowledge about the diversity, right? Now, genetic potential to gene expression. We have seen that the genes by the transcriptomic analysis and the expression from genes to transcripts and from transcripts to gene expression, both the two things, this is what I have mentioned already. So the pure presence of microbial groups which can carry out a given function in soil cannot be used as a proxy for potential microbial function. Means you cannot be 100% sure that this is a proxy that you getting certain specific function means that function will be always available. It is not there. So also the soil proteomics also. So this is the transcription to gene expression of the gene. This can give us the mechanistic understanding of how soil microorganisms respond to changes, different changes. And that can be found out, as I said, that the anthropogenic impact, like cultivation, you are practicing cultivating the soil, we are putting water into the soil, you are spraying pesticides and other type of things. There is the impact of the drought, all sorts of things, anthropogenic and natural impact. They can influence the microbial activity into the soil. Now, what is the real value of soil biodiversity? Why should we know it? I will give you 2 examples which will tell you why it is so important, apart from other things. Let us take for nitrogen fixation. It actually saves around 70 billion euro. You can convert it into rupees. How many 1,000 crores, 100,000 crores of Indian rupees will come into it. The nitrogen fixation, around 70 billion euro, is the potential of the nitrogen fixation by the microorganism. Pollination, of course, not only microorganisms, there are also some larger insects are also involved in that, and that contributes to around 150 billion euro. So you can see here that the soil biodiversity has got a large economic role to play. So now, why we need to protect biodiversity? I have given 2 examples of economic value. So economical or utilitarian reasons, then ecological reasons and ethical reasons. Ethical reasons means you go to the forest, look at the forest and be very happy that it's a nice serene atmosphere, where the plants and the soil and the environment are pristine, and then you get peace of mind. That is the ethical impact. And the ecological reason is that the different soil, different ecosystem are functioning together, and those are the ecological reasons. So these are the reasons that we need to protect soil biodiversity. Three main reasons are the economical or utilitarian reasons, ecological reasons, and ethical reasons. Now, I'm talking, just mentioning about the ecosystem services, which you might have studied in the ecology portion of your disciplines. Now, if we take up the soil biodiversity, there are actually four ecosystem services, major ecosystem services it support: provisioning, regulation, cultural, and supporting. Provisioning means it provides food. Soil biodiversity, supposing the BNF or the biological nitrogen fixing agents, they are there. They help into the fixing of the nitrogen and supply it to the plant. So the plants grow and then we eat that crop, so providing the food. In addition that, the group of microorganisms, they serve as a food for the organisms. Then the fresh water, then the wood and fiber, obviously forest produces and other things, and fuel. So this is the providing resource, these things to people, or the ecosystem services, that is the soil biodiversity supports, that is the provisioning. Regulation, climate regulation. We are nowadays, we talk about the climate change and all those things. Now, these microorganisms, they actually help in the regulating the climate. You may ask how the microorganism, a small bacteria in the soil, will result into the climate regulation. Yes, it helps into the sequestration of carbon, carbon sequestration. You know, carbon dioxide is a major greenhouse gas in the earth and result into the warming of the climate. Then the different greenhouse gases, methane and nitrous oxide. Methane can be mineralized to carbon dioxide by the microorganism, and the other nitrous oxide. Nitrous oxide may be reduced to nitrogen. So this way they help in the climate regulation. In fact, I mean, if you forget about the Eocene era, Anthropocene era. Before Anthropocene era, the climate, the ecosystem, especially the nature and the environment, they are all in equilibrium, and they but these microorganisms, they can help regulate this thing. They can help the they can help in the disease regulation by allowing the resulting into the creation of the antibiotic formulation into the soil, so that the microorganisms are lost. Then the biological control type of organisms are there, like trichoderma in the soil, which controls the growth of disease causing bacteria. Then use erosion control. The bacteria, they may control the erosion of the soil by forming large clumps resulting due to formation of the macromolecules like polybeta hydroxybutyrate, resulting into the clogging of the soil particles, and thereby they may control the erosion. So this is actually the regulation. Cultural, I have mentioned about this: educational, spiritual, aesthetic. Excuse me? Yeah. So the supporting is the decomposition of organic matter. These are the supporting services. Decomposition of organic matter. So this will, by decomposition of organic matter, this may lead to the providing the food. Then the nutrient cycling. Nutrient cycling is the resulting into the growth of the plants, soil formation, and the primary production. So these are the biodiversity and ecosystem services. So soil biota moderate primary production through their role in soil formation, decomposition, and nutrient cycling, control of or suppression of plant pests and diseases, production of plant growth hormones, the regulation of water supply by modifying soil structure and water filtration and storage, and promotion of flood and erosion control by modification of soil structure and water filtration. So there's a huge gamut of functions the soil biota they support in order to moderate the primary production in the ecosystem. In addition, the soil biota also influence climate through, as I was mentioning earlier, storage of carbon, what we call as the carbon sequestration, production and consumption of greenhouse gases. I have given the example of the methane and nitrous oxide. Methane can be oxidized to carbon dioxide. Nitrous oxide can be reduced to molecular nitrogen and converted into harmless thing. So the scientists and policymakers are increasingly recognizing the benefit of including ecosystem services in decision making and global sustainability. Now, I just take a case study of a soil microorganism affecting agriculture ecosystem services. I mean, probably you know all these things, but it is shown in here in a picture by Scow et al. 2013 picture, but I liked it very much, where you can see here that this is a typical ecosystem, agricultural ecosystem. You have got the crops growing here, in this portion. Then you have got the forest, all the forest trees. You have got the industry, the factories are there. And then you have got the villages and the all sorts of things. And these are the different, and the wild areas, where the rocky and other type of things are there. Now, in this case, what happens is that the breakdown of the wastes and make compost. If you take from the which is not natural, let us start with that, like factory. In factory, the different wastes and other things are produced, and the microbes can break them down and make compost. Development, now the effluent, they will result into the several antibiotic resistance they may come, and that can be controlled to break those things. Here also the same type of those things also, those compounds can also be broken down by the organisms. It is here, biodegradation, etc., etc. The forest, actually, they support this plant and animal via mutualism. The forest, they support the birds to roost, and also the leaves will fall into the soil, which will result into the building up of the soil organic matter, and also the control and cycle the plant nutrition. This soil organic matter is then used, serves for growth of the crop. You are growing the crop in the cultivated field, and then they will be used by this growing crop. This organic matter will result into the increase in the fertility of the soil. There are also microorganisms which will fix nitrogen and then allow the crop to grow. At the same time, they can control the plant diseases and etc., etc. So here different type of pests and diseases, they are controlled through this integrated pest management, different through microbial or microorganisms, especially as well as small animals like trichogramma, etc., etc. Those are they can they can destroy the pathogens, and they result into the protection of the crop. The pesticides and other things which are harmful chemicals that are sprayed for protecting the crop as a short-term measure, they will ultimately end into the soil, and they might affect the soil biodiversity. And here the microorganisms, they will break down the insecticide and pesticides and result into a more harmless environment. So you can see here, these microorganisms, they have got multifarious roles, starting from the building of the soil organic matter and transformation of the nutrients to all sorts of things. I have not mentioned this here, where the what that called, the soil structure, the virgin land, the rocks and other things, those are broken down by the microorganisms, and ultimately the soil is formed. These soils, on these soils actually you grow the crop. So this is process also microorganisms are involved in that, and the whole thing thereby contribute to the biodiversity. So you can see here that the interaction of the microorganisms into the ecosystem is a multifarious, but resulting into the protection and help of the protection of the biodiversity and helping the human being. Now, I show here the four major main ecological concepts influenced by the soil biodiversity. These are the four pink, this thing. One is the resistance and resilience, ecosystem adaptability, then the current and future ecosystem service provision. We have already introduced these things to you earlier, as well as the new biotechnology projects. Now, these are all supported by the soil biodiversity, because at the top you have got the soil biodiversity. They will result into the pool of life history traits and functional characteristics. These soil biodiversity, or the organisms which are responsible for maintaining the diversity of the soil, they help in the formation of a pool of traits and functional characteristics. Now, there are that is all. These things are then moderated by the soil processes and the reservoir of the novel genes in the soil. The moderation of the soil process will lead to the resistance and resilience, as well as ecosystem adaptability, and the reservoir of the novel genes, new genes which are present into the soil, they can help into the formation and production of the new biotechnology products, and then together they all current and future ecosystem service provision can be obtained. So coming towards the end, I tell you here the soil biodiversity moderates soil processes such as nutrient cycling, and through this directly influences the provision of ecosystem services. Soil biodiversity also influences the resistance and resilience of functions of perturbations such as pollution and extreme events. I have mentioned the biodegradation of the pesticides and other things which would otherwise have polluted the soil. So these are the and also extreme events like say for example drought. A drought is there, means there's not the microorganisms present in the soil will result into the formation of the soil clump, so that in a dry soil the erosion is protected. So these are the different ways that the soil biodiversity influence the resistance and resilience of functions. Moreover, soil biodiversity provides a pool of novel genes. The earlier picture here, the novel genes, reservoir of the novel genes. These novel genes actually, the genes cannot come into the soil like that. The other, where it was residing, the microorganism, after their death and decay, those DNA fragments come to the soil, and this reservoir of the novel genes, they can help into the production of new biotechnology products and then improve the, so together the pool of novel genes that may improve the ecosystem adaptability, but also represents a significant reservoir of genetic material that may be utilized for biotechnological products in industry. So you can see here, that means there is a very, the diversity, it has got a multifarious actions into different way, and then they help in the maintenance of the soil processes. Now, here you can see, this is, I am just sorry, this is the FAO they had the decade on biodiversity 2010 to 2020. Now the conservation of sustainable use of soil biodiversity, they are specifically listed them. They are already, I mean, these biodiversity program, soil biodiversity pool, it was already in existence and government endorsed the main international framework of action regarding the biodiversity. Now, the managed mainly by the APO and other partners, these multinational organizations, they have got a different goal of awareness raising, making people aware about maintaining the soil, knowledge and understanding, and mainstreaming. Then the strategic principles is that improvement of farmers' livelihood, irrigated, adaptive, holistic, and flexible local solution. Participatory technologies can develop suitable development suitable to local conditions. Then building partnership and alliances, promotion of cross-sectoral and integrated approaches, and then finally the dissemination and exchange of information and data. So these are the different managerial options that actually FAO supports while conserving the sustainable use of soil biodiversity. Now, this is a global soil partnership which is shown here, this is a partnership which actually these are composed by partners who actually they advise the intergovernmental technical panel on soil, and then you have got the Global Soil Protection Secretariat, GSP Soil Partnership Secretariat, and through the regional soil partnership, they actually support the biodiversity research. This, I think, I need not to, I am not going to tell field scale. From the field scale to ecosystem scale, I was telling you field scale. So here is a small field of crop growing, and then you have got the increase it to the farm scale, where the large scale of action is there. Then this farm scale can be further increased into land scale basis, whereby you can show the entire continuity of the diversity, soil biodiversity, microbial biodiversity from the field to the landscape scale. Now, these are the big different things which are mentioned, but basically you can see here, if you see the benefits of the soil biodiversity, is that nutrient cycling and uptake, nutrient and water use efficiency, plant growth, health and stress tolerance, then also pest, pathogen and disease suppression, then the soil structure and weather details. So these are some of the things which are, I mean, they are at the field level. Now, when you go to the field level, to the farm level, in place of a small, a few square meter field, to a farm level, there could be several hectares. They support the local population of beneficial organisms. In the farm section, on the boundaries, plants are big, I mean, trees are there, bushy type of things. They help the other organisms to thrive. So, and also, they support the local, I mean, improve the species diversity, including antagonists of plant pests and pathogens. Then once you can go further up, that is the that's the landscape basis, where the large, huge area is shown. Now, this the regional species and species pools from which the beneficial diagnostic organisms can disperse, then improved broad-scale provision of services, and adaptability, resistance, and resilience of environmental change. So you can see here the soil biodiversity, they control at different levels of analysis, they control the at different production system. They can go from the soil, field soil, to the farmer's field, and to the ultimately to the landscape, so that we find that they can benefit the production system at various spatial scales, because related to space, from the field to the farm to the landscape, and how management practices at the same spatial scales may accentuate the potential benefit. I think this is the last slide. Yes, this is again another poster: protect soil biodiversity, keep soil alive. If it is, we have already probably used it to a great extent, but then now the pressure is on you that you should protect the soil biodiversity and then leave a more healthy world to the next generation. Thank you very much. Yeah. Maybe I will wait for questions, or how is the procedure?
Very nice presentation, sir.
Yeah, thank
Very
you.
informative session. Thank you, sir. Dear participants, please ask the questions. Unmute your mic, ask the questions. Otherwise, post the chat box. Dear participants, unmute your mic, ask the question. Otherwise, post the questions in chat box.
Sorry.
Good morning, sir. Sir, you have explained. Chat box. Sir, use chat box now, sir.
I think some echoing is going on. I'm not sure.
Please post on chat box. Otherwise.
Yeah, that will be nice. I can see you're posting in the chat box and reply.
Dear participants, kindly post on the chat box.
Yeah, question has to come in the chat box.
No questions. Sir, one question in chat box.
Yeah.
What are the immediate fallouts of soil acidification for the survival poor in India, and how in inequalities are exacerbated?
Yeah, I think it's a good question based on ecosystem aspect, especially the soil desertification. Uh, Desertification is because of the, obviously, you know that the lack of moisture is the first trigger, and then finally the plants are not able to grow, or due to some other problem in the climate, that the desertification takes place. There are several reasons, let us not go into that. But then obviously the microbial biomass or the microbial diversity in those type of ecosystem gets changed. It has been seen that the diversity, soil biodiversity is changed even because of the minor, as I mentioned earlier, natural or anthropogenic interventions. Now, taking into consideration, I mean, thinking that the desertification is a natural intervention into the ecosystem, then the microorganisms also adapt themselves. And then they can allow, as I mentioned, immediately desert. One of the problem will be the retention of moisture. Then there could be the erosion of the soil. Then there could be these two are major. In addition, there will be several other things which can help happen in the desert ecosystem. Now, coming to the moisture retention, the microorganisms, they produce different polymers like polybeta hydroxybutyrate, etc., which this results into the formation of a film around the soil particles to retain water, so that the to the extreme harsh condition of the water availability, that these microorganisms or the soil microbes, they can affect. Now, the another thing is that soil erosion. Usually desert areas, the crop is not there, plants are not there, the large areas are open, and because of the wind, the soils can be eroded and then get deposited into another area, or I mean, that's how it is lost. This is a great problem, big problem rather. And this also microorganisms can help the soil by forming clumps, so that they are not eroded, and they are all at the same time helping the other plants to grow naturally in those soils through retention, as I mentioned, retention of moisture and the avoiding the nutrition. And apart from that, there are other provisioning services like nutrient cycling, etc. They can help the plants to grow, thereby restrict desertification to a certain extent. Now, if we are to do on a large scale, of course, then the normally, I mean, it has to be seen what are the reasons, what are the group of organisms growing there, and what are the types of plants that can be grown, and thereby this entire harmful effect of desertification can be excavated. Okay, is it all right, whatever I mentioned?
Next question, sir.
Yeah.
Which method is thought for estimation of microbial biodiversity?
This is a very interesting question. As I mentioned, that no single method is perfect. And then you have to take the help of several methods. One of the commonest method we use is the for measuring the microbial diversity is the microbial biomass estimation. Microbial biomass estimation can help us to know the amount of biomass present in the soil, and then also at the same time, by calculating back the nitrogen, phosphorus, and carbon content of this microbial biomass, we can predict the amount of nutrient transformation that the microorganisms can help. So this is a basic method for measuring the diversity. But as I said earlier, this no method is a sacrosanct, and then one method is not possible. There are different methods. I have mentioned a few of them. You can check with the DNA content of the soil, of the sample, and like that several other methods also can be seen. But one of the commonest is that microbial biomass estimation. We can find out what is the extent of microbial biodiversity. All right.
Yes, sir. Any questions from participants? Any questions? I think no more questions, sir.
Okay then. So no question means that's fine. I mean, as I mentioned, that microbial diversity is one of the interesting content of the ecosystem, and that helps in several ways to maintain the ecosystem harmony and result the plant and the animal, and also the largest animal, that is the human being, to stay in the ecosystem in a harmonious way, and thereby maintain the natural ecosystem surface, ecosystem activity to a great extent by involving this biological diversity, especially that of the microbial diversity. Thank you very much then.
Thank you, sir. Thank you very much.
I think I will meet again at 1:30.
Yes, sir. Yes.
Okay then. Thank you very much. So I quit. Yeah.
Yes.
Yes, sir. Yes, sir. Thanks for your availability, sir. Thanks for your
Okay.
availability. You have to
Okay.
be in the afternoon session, sir.
Sure.
Around 1:30 after you, sir. Okay, sir. Okay. Thanks for your
Thank
you
you.
very much.
Thank you.
Thanks a lot. Thanks.
And this is the same link will be there for evening
Yes,
afternoon
sir. Yes,
session
sir.
also.
Same link, sir.
Okay. Okay. Thank you very much.
Thank you, sir. Thank you, Ajay. Thanks for your Shiva, sir.
Yes, sir.
So the session is wind up for that morning session. We have some break now, as the physical audience have to take some refreshment. Then we can again
11:10.
Exactly at 11:15. Better you can join at 11:15.
Dear participants, again you can join at 11:15. Thank you. Thank you.
After studying your financial concepts, now we are studying your
Shyam, Jumail. Shyam or Jumail.
Yes, sir. Hello. Shiva, sir.
Yes, sir.
Okay, so we can leave the students for break.
Yes, sir. Yes.
Next session will start at 11:30. Yeah.
11:15, sir.
11:15.
Yes, sir. Sharp
It's not
start at
possible.
11:15.
Okay, okay, no problem.
15 minutes, sir. Dear participants, next session will start at 11:15. Thank you.
Zip.